Good evening, everyone. My name is Rob Saint, and I'm the Pro Vice-Chancellor for Research Strategy at the University of Adelaide. And I have to say it's an enormous pleasure to be here in this wonderful new lecture theatre in this incredibly beautiful new research building here at the University of Adelaide to introduce one of my very close friends, Professor Simon Koblar. Simon gained his medical degree in 1984, I believe, and then specialised in neurology and worked, for example, at Guy's Hospital in London, as well as here in Adelaide in that specialty. And then for some very strange reason, I'm not sure what possessed him, he decided that medicine wasn't enough, that he wanted to be a researcher as well, and he undertook a PhD degree at the Walter and Eliza Hall Institute in Melbourne, I think around 82 to 86. And followed that with a postdoctoral period of research at the University of Washington. I knew it was Seattle. I was guessing it was the University of Washington. So Simon's a somewhat rare breed of person who has embraced both the clinical side and the research side of medicine. And he's done that incredibly successfully, as you'll see from the talk. And he continues to combine both of those. He's got a number of titles. He's currently the Professor of Medicine here at the University of Adelaide and also at the Queen Elizabeth Hospital. He's the Clinical Advisor for the Centre of Stem Cell Research in the Robinson Institute, and he's Director of the Stroke Research Program at the University of Adelaide, Queen Elizabeth Hospital, and the new South Australian Medical Health Research Institute, or SAMRI as we refer to it. So he's a very busy person. He's been enormously productive. He's been a great mentor to many PhD and honours students. He's won many awards, so many I'm not going to list them. He's funded by the most competitive funding schemes in the country, the NH and the MRC, and on top of that, he's one of the most enthusiastic and wonderful people I've ever met. So it's a great pleasure for me to introduce Simon. I'm really looking forward to hearing his talk, and I'll turn it over to you, Simon. Thank you.
Thank you very much, Rob. Welcome, everyone. To a number of people that I know. I'm hoping that there are more people I don't know than I know, or else it would seem as though you've invited people to your own party to have a good time. So tonight is, I thank everyone for being here. Tonight is particularly important because it is the first time that I'm going to actually suggest a study that's hopefully we'll be able to achieve here in Adelaide. And many times people say to me overseas, where is Adelaide and what's special about it? We all know why it's special, but this I think will be very important. So I'm going to talk about a range of neurological diseases. I'm a neurologist, as Rob said, and I see a range of patients. I'm a general neurologist, but also I have a particular interest in stroke, and so I'll be focusing on that in the latter part of the talk. So we tend to do this now where you can see what people's affiliations are and if they're biased. I'm very biased in the sense that I have been funded for a number of years by the Couche Foundation, which is part of the Robinson Institute and part of the University of Adelaide. And more recently, I've had interest and formed a collaboration with a company, which is an Australian company called Mesoblast. And you'll see why that's important in regards to actually taking something from the bench to the clinic, which ultimately is a test of whether something is going to be worthwhile. And obviously, there's a great hope in regards to stem cell therapy. These are the people that do the work. A number of them are sitting there in the middle of the row to my right, your left. And this was a particularly good occasion because they all came along. It was just around Christmas time because I just secured this relationship with Mesoblast, and I shouted them to lunch just down the road by Jolly's Boathouse. So it was a particularly good occasion. They look very happy because that's the end of it. So tonight I'm going to talk about 4 areas. And And then at the end, I'll have 3 microphones, and some of the more athletic in my group will run around and give you the mics to ask questions. We'll do it in that manner. I'll be talking about stem cells at the beginning and put you in a picture of what a stem cell is from a biological point of view, and hopefully make it reasonably sensible. Then I'll talk about some concepts that I've gained an appreciation for over the last 10 years in regards to where stem cell therapy might lie and what it might actually do. Then some very exciting work around the world in regards to clinical trials that are up and running now and will be, and also about a terrible thing of stem cell tourism that I'm sure a number of you are aware of. And then finally, I'll be biased and spend probably half of the talk on my favourite stem cell. Everyone has their favourite stem cell because it takes you a lifetime to learn a stem cell. And mine is from the tooth, and you'll see how that transpired and where it's taking us. So Rob Saint didn't tell you, but he actually gave me my first laboratory here at Adelaide University many years ago, and he took a great gamble because I was a clinician trying to pretend I was a geneticist, and he actually taught me a love of embryology and the genetics of it. The word stem cells is a relatively new and sexy term, but in fact we know it as embryology, modern embryology. And in fact, it's built on a foundation of at least 200 years for the modern embryology, which equates to the first microscope, so being able to see these small organisms. And this is a lovely picture. I'll just project onto the right, on your left, so follow the red dot. So actually this is a drawing from an old embryological text and showing a human embryo from its early stage through to gestation, and a whole lot of different organisms, right down to fish, and I think we've got pig and various others. And you can see the amazing similarity between these organisms at different stages during their gestation, whilst they're in the womb and the development. And so the first concept that was gained over the 18th century was the morphology, just the changes that happen to the embryo, which is quite amazing. There's this incredible period of growth to actually start at one fertilized egg through to actually having this complex organism that in the main generally is born with pretty much things intact and right. We then added on labels to all of these over the last 200 years. Rob Saint worked with a fruit fly called Drosophila, and I've worked with fruit fly and zebrafish and rats and mice and not primate, but humans. So each of them has their own specialty and their own ability to teach us more about embryology and how this fantastic development occurs. And we've realized that there are growth factors and various things that are critical at different stages and different steps to build this organism, and also that there are genes that direct them, and that there are programs of genes, and that there are actually, we now recognize networks, and there are some master genes that coordinate how an embryo is developed, and then others that are slaves to those master genes. So with all this knowledge, we then came to probably the 1990s when many of us in embryology started asking the question, how can we translate this? How can we move it from what we know in the laboratory to a clinical setting? And that's where stem cell therapy and the concepts were born from there, how to apply this great deal of knowledge. So what is a stem cell? A stem cell is basically a primitive cell, and it starts having two major features. One is that it can continue to proliferate or grow and multiply. And in fact, Jeremy Thompson, who made the first human embryonic stem cell, has a cell human embryonic stem cell that's growing in his laboratory in America has been for the last 30 years. It also has ability to make every cell type in our body. So there are 264 different cell types that make up the human body. And if you take an embryonic stem cell, which comes from a very early stage, from a thing called a blastocyst, where there are a number of cells and you can't recognize any human from that, you can produce these continuously going embryonic stem cells, and they can make everything from a brain cell through to a skin cell through to a liver cell. And then what we've recognized over the last 30 years is that in fact almost every organ in our body has a reservoir of stem cells that reside there. And often they're very small numbers. The brain has some stem cells. So there was a dogma that as soon as you were born, from that time you just lost brain cells. So if you had a hard life, you lost them greater. And we now know that, in fact, that's not true. There's an ability of the brain to actually regenerate to some degree, and I'll talk about that later. So there are adult stem cells that are found in different organs. And then last year, Yamanaka from Japan won a Nobel Prize for an incredible discovery, which I'm sure Rob and I never saw coming, even though we had all the tools there. He took a skin cell and put in 4 genes and transformed that, reprogrammed that skin cell back into, in all intents purposes, an embryonic stem cell. And that's fantastic. And in fact, I was at an international stem cell conference last year, and Japan are putting in enormous resources to have this concept of individualized stem cells. Each person, you can take your own cell and make this stem cell, and perhaps that can be useful for regeneration and repair in the future. But that still has a number of issues associated with it, which I'll talk about later. So then the concept is that this unique cell, this stem cell, is able to regenerate tissues and organs and perhaps be involved in repair. I'm going to spend tonight mostly talking about a population of stem cells called adult stem cells. And in fact, many times we forget, but hematologists have been using stem cells from the bone marrow for the last 30 years. And so we have a great deal of knowledge, a great deal of ideas about the safety and perhaps not safety of stem cells from the bone marrow. We know, and I get a number of phone calls or emails from people saying when their baby is born, should they store away the umbilical cord cells for future use. So that's another very common stem cell population which is used in different studies. As I said, there are stem cells in our brain, even at my age and older. We know that they're involved in some repair processes. It was probably too limited and too late in many ways. There are, in fact, stem cells in fat tissue, and you might say, well, why would you need to do that? I don't need to become fatter, so obviously not a great deal of need for that. But however, it gives you, you might think, a ready supply of some stem cells. And finally, I'm going to spend my time talking about teeth and the stem cell population that grow from there. So, stem cell therapy. There are basically 2 ways of thinking about how stem cell therapy might work to repair damaged nervous systems, be that whatever type of nervous system disease. One is endogenous. So I'll use some hard words, I apologize, but I'll explain what they are. So endogenous just being inside the tissue. So inside the brain, we know that there are stem cells. And in fact, there have been a number of studies on a number of different diseases, from Alzheimer's disease through to Parkinson's disease, where we know in humans when the people have died and we've looked inside their brains, that there are stem cells that are trying to repair some of these neurological diseases that damage our nervous system. And many of you know all about these particular diseases. However, as I said, the response is not enough. to make an impact on the person's life. And so the major question in regards to this particular idea of endogenous repair is, how do you increase that, the number of stem cells in your own brain? And we know that in fact exercise is a wonderful way of actually improving your number of stem cells. There have been a number of studies in different animals to demonstrate that, but obviously if you're in a wheelchair, as a number of these people at the front are, it's a little hard to exercise. So there are a few limitations in that approach. We know that there are a number of growth factors, and in fact my old PhD supervisor Perry Bartlett was here last week and said that they think that growth hormone, which is a hormone that helps our adolescents and children grow, might be very fundamental in regards to increasing the number of neural stem cells in the brain. And there are medicines that we know now actually have an impact on increasing neurogenesis or neural stem cells in the brain. And one, interestingly enough, is our antidepressants. And we know that they actually have an impact on a part of the brain where there's a stem cell population. And so the idea is how to utilize different ways of increasing that over time, but obviously doing that in a particularly targeted manner, not one that will give you other side effects, and sometimes that's the biggest problem. Now the other approach, which is the one that I'll probably concentrate on today and really relates to most of the clinical trials that exist around the world, is in regards to exogenous treatment. That is taking stem cells from outside of the brain generally to be used for therapy. So this often requires transplantation, and it can either be through the brain or the vein, like that. I like that, catchy. So you can either inject it directly into the brain, or you can give it via the vein, intravenous. Now there are two ideas here of how to do this exogenous stem cell therapy. One is taking a stem cell from somebody else, which is what companies in this field generally do. They'll develop a stem cell of one particular type from a particular human being and have that multiply so that they have a product that we can, as we say, buy off the shelf. The other approach is what's called autologous, or your own stem cells. And that is where you might take a tissue from yourself, such as a sample of the bone marrow or your tooth that I'll describe about, and then give that back to yourself. And obviously that's a much safer mechanism of stem cell therapy in the long run. But obviously sometimes that's not always available. And obviously the best stem cell for neurological disease would be neural stem cells, but we generally don't like to give up our own brain for that, and that is too risky a procedure, so that's a limiting factor. One of the biggest problems of stem cell therapy, particularly for embryonic stem cells and indeed induced pluripotent stem cells, are tumour formations. And in fact, there was a very tragic case of a young boy who was 15 years old from Israel who had an awful degenerative disease of the nervous system. This was reported some years ago. And he was the first example of someone that had been transplanted with two different embryonic-like stem cells, and some years later developed tumours throughout his nervous system. He'd gone to Russia and had paid money for a particular clinic to give him this. And so this was the first human evidence that, in fact, what we'd seen in animals, that embryonic stem cells could actually result in tumours if they were put in the nervous system. So how do stem cells work? So this is really, Yamanaka won the Nobel Prize last year. This is the next question and challenge for the field for a Nobel Prize, in my view. So there are a number, at this stage in 2013, we believe in the field that there are probably 5 different ways of approaching, of thinking about what stem cells might do. And I'll concentrate here particularly on the nervous system in the next slide. But I'll just go through each of these. The simplest one to consider is a stem cell might replace a lost brain cell or a particular cell that's involved in a particular disease. And this concept of replacement really comes from hematologists, because when someone had leukemia and you gave them chemotherapy and you wiped out all their their blood cells, then you gave them a transplant of stem cells, either from themselves, from previous harvesting, or from somebody else, and so that replaced what was lost. And this was really the major concept that was considered for stem cell therapy of a mechanism. But we've now recognized over the last few years that there are a number of other ways that stem cells might play roles, particularly in the brain. One particularly important one is a thing called immunomodulation. So many diseases of the brain involve inflammation, and Stefano Placchino, who I've worked with in Cambridge and still do, he was demonstrated in the early 2000s that in fact neural stem cells could actually regulate the immune system and change the immune system. And so that was a wonderfully new and exciting way of thinking of how stem cells could change neurological diseases. The other is in regards to angiogenesis, which is a fancy term for making new blood vessels, and sometimes that's important, particularly in stroke. Neuroprotection, so Josh has put this shield up here to show you that you're protecting the brain. So basically these stem cells can be considered as little growth factor factories that produce these chemicals that can do a whole range of things in the brain. And the final thing, which I'll talk about to later in a bit more detail, is called neuroplasticity. So there's a concept that in the nervous system, it is not hardwired. It is not just static and doesn't change. I always say I've studied and in love with the brain and the nervous system. And basically, I think the biggest insult to the brain has been people comparing it to the computer, because the computer is such an inferior thing compared to the brain. This brain can change. If it weren't able to change, you wouldn't be here, because you're here learning and hoping to gain knowledge about this. So if it was static, it wouldn't change. In fact, I'll show you later in some wonderful imaging that the brain is actually, after a stroke, able to change where it's able to function. So there are these 5 mechanisms that are considered. So I'm going to talk about a number of neurological diseases, and that's why you're here, and where we're up to with regards to some clinical trials. Let me talk about some to do with neural replacement. So there's 2 particular conditions, Parkinson's disease and motor neuron disease. And both of those diseases occur because there's a particular type of brain cell or neuron which is lost. So for Parkinson's disease, it's a brain cell that produces dopamine. So you might imagine that stem cell therapy would be about replacing that lost dopamine neuron. Motor neuron disease is an awful disease where the motor neurons actually die, and so you become weak. And so to replace those motor neurons is a key element, perhaps, in stem cell therapy. Spinal cord injury and stroke might require neuroplasticity, this changing nervous system to help overcome disease and improve function. We know multiple sclerosis, as we call it, an autoimmune disease. We know that it's due to, we don't know what produces it, but we know that it's due to an overinflammation within the nervous system, and in fact immunomodulation or changing the immune system might be critical in regards to how stem cell therapy might work in this regard, in multiple sclerosis. So the final one, say neuroprotection, can have a role in a number of these different diseases. So the other concept that I've realized is important, and I used this a number of years ago for my colleagues, doctor friends, because they couldn't understand when I was doing the very early work on this in embryos what relevance it had. And I've recognized over the last 10 years is that we often think about medicines. If we take a tablet, then it's generally a one-way interaction there. So you take a drug for your brain, and that has a one-way action. The unique thing about these stem cells is that they're living cells, and that there's actually a two-way talk that occurs, both of the stem cell through to the brain or nervous system, and the nervous system through the stem cell. And I'll show you evidence of that, not in the human brain, but in other organisms that will help us understand some of that. So I'll talk about clinical trials. So some of you might be aware, but every time you take a medicine, then it's generally taken 20 years for that to get to the clinic to be able to be used. And $1.5 billion, whatever that is. So it takes a long time and a lot of involvement. And in fact, 99.99% of all discoveries that scientists like me get very excited about fall by the wayside because of a whole range of things that just don't work to mean that they can't come to influence my clinical practice. And there's this wonderful concept called the Valley of Death, where in the pharmaceutical industry they say that everything falls by that wayside. And this is from a lovely picture from a recent review from the States about this. I put the star there because I think that's where stem cell therapy is. It's at a start of clinical trials to see whether it's safe. You'll see most of these studies. Not whether they're able to be beneficial and improve function at this stage. And then really, as much as some doctors and a number of people might be against pharmaceutical companies, they're fundamental in getting medicines out. And so they have to be able to see that that's going to be useful, it's safe, they're not going to be sued, and that it's going to actually make a profit for them, which may not flow through to anyone but their shareholders, but that's life sometimes. The other important thing is there's a particular, although I have many of my patients say, why can't we go now? And this occurs in cancer treatment and infections such as HIV. There is a process that's developed over the years, and it's really one that I think has good sense. And that is basically for safety and to prove that things are worthwhile. And there's certain phases of clinical trials. There's a phase 1, which we tend to start with, which is basically to prove if a treatment is safe. And often you use small numbers of patients. Why do you do that? Because if I'm a clinician and I'm treating people with something new and someone dies, I feel terrible. And it's obviously tragic. And so you don't want to have that happen to a large number of people. So you start that in a small number. Then you have phase 2 studies, which again is to do with safety, but sometimes changing things to look at the benefit. You get to phase 3, which might cost you $500 million to mount these studies, and often they are what's called randomized control studies, where you have people given a particular treatment and those that are given not the treatment, but they think that they might be, so it's what's called placebo-controlled. And you might do that for many thousands of patients across the world in many studies, because we know that there is bias in every trial that's done. And the best trial to do is to unbias it outside of one particular environment, because we all have vested interests. And then finally, phase 4 is when medicines become on the market and patients and doctors give feedback on how good they are or what side effects there might be later. Post-marketing, we often call it. So I'm going to now talk about these diseases and just outline some of the aspects of where things are in regards to clinical trials. So I've put this in order of the number of patients that I see with these conditions. So there's a general type of trend here. So multiple sclerosis is a very common disease, and unfortunately, and there's a number of different flavors of MS. One is a thing called relapsing remitting, and in fact, there's a number of fantastic drugs that are available now, medicines, that can treat that very effectively. And I'm old enough to remember when we wouldn't even tell someone they had the diagnosis because there was no treatment available. And now that's all changed. The big problem is that a number of people, maybe 20, 30% of people with MS, have a progressive form of multiple sclerosis. And in those cases, those people, there is no treatment that can be used. And this is where stem cell therapy is a possibility. One, if repairing the nervous system that's been damaged in MS, and perhaps involving stopping the continued progression. So I have a number of colleagues in Cambridge. I was fortunate to be there in 2010 on sabbatical. I do a number of sabbaticals, as I've told Rob, and which is study leave. It's actually working. Alistair Compston's the head of neurology at Addenbrooke's Hospital, and he was involved and set up this fantastic study, what's called a phase 2 study, which was taking patients, very clever study, taking patients that had progressive multiple sclerosis, so they were deteriorating, but where they particularly had visual problems, a loss of vision, so they were blind generally in one or two eyes. And all they did with this study was just to look at their vision and their optic nerve that gives you vision. And so it was a very focused study. They worked with their hematologist colleagues, and they took bone marrow samples from those patients, isolated a stem cell from the bone marrow and gave it intravenously or into their vein back to them, one treatment, and followed them up to 10 months, There were 10 patients, and there was a range of ages, I think generally about 40 to 50 years of age. And the major outcome that was looked at here was safety. And so the first aspect is that they were able to demonstrate and published at the beginning of last year in a very high-ranking journal in the world that this was safe, a safe treatment. The second is that it's called a phase 2 study because they actually looked at whether it might be of benefit. But the problem is that 10 patients is too little to say anything really about. That can all be placebo. That is, I give a sugar tablet to someone and say, this will cure you, and if I am very convincing, as often I am, then they will believe that it will, and it will help them. However, they did demonstrate that there was an improvement in vision and a number of things to do with this optic nerve over the course of that 10 months, which was quite remarkable. So following on from that, and I don't quite know why they didn't continue on at Addenbrooke's, well, I actually do. I talked with them about that, and basically the person that had set this up took 10 years and he's now retiring, so he couldn't take it to the next phase because it takes a long time out of your life. So this MESIMS is the next study, and this is being run by Bertolucci out of Uccelli out of University of Genoa in Italy. Just started, and again it's another phase 2 study, but it's supposed to be much larger, perhaps 100 patients or more. When you go to the website, there's a clinical trials website, and look, at this stage it says they're only recruiting 20 patients, but I'm in discussion with the group from University of Genoa, and seeing whether they're actually doing a number of centres around Europe and also involving Canada, and seeing whether we might actually be a centre here in Adelaide for this particular trial. And again, this is autologous bone marrow transplantation. So it takes your own stem cells from a patient that has progressive multiple sclerosis and gives it back to them. And it's supposed to be looking at safety as well as benefit. The next is Parkinson's disease. Parkinson's disease is, or even though everyone thought it was maybe the easiest disease to treat because you tended to have a loss of one particular population of brain cells in the brain causing this disease, it's actually ended up more difficult than some of the others to get started. In fact, there are very few trials. There are 5 human studies that are currently registered that are being done around the world. Most of these studies are using tissue from aborted fetuses, a particular part of the brain that actually makes the brain cells that form the dopamine that is lost in Parkinson's disease, which is all that terminology there. There's also other studies which I think have little evidence that they will be of any benefit, and that is using bone marrow and adipose or fat tissue in various other countries. One of the biggest pities with this is that in 1992, a neurologist in America, Freed, started using these particular fetal cells, tissue, and transplanted about 40 patients with these and didn't demonstrate any improvement. In fact, demonstrated a side effect that no one expected, and that is where the patients developed a whole lot of extra movements that were quite debilitating. The biggest challenge actually for the stem cell field is not just making a neuron that has dopamine or produces this chemical in the brain, which is fundamental for Parkinson's, but something that actually is able to integrate and sense the loss and actually be able to deliver dopamine when it's needed. And that's the biggest test. And when I was in Boston just in June for the International Stem Cell Society meeting, we still hadn't achieved that using all of our technologies and all the different stem cell types, from embryonic through to induced through to different adult forms. So that still is a work in progress, even though for many of us we thought that that might be the first disease that would fall. Spinal cord injury gets a lot of press. From my perspective as a neurologist, it's probably a less common disease than many of the other diseases, and people are fully aware of Christopher Reeves and all the great work that he did to bring this out to the world. Currently, there are actually 14 human trials registered, 3 completed without any reports. They use a range of different stem cells, but you can see 10 using bone marrow stem cells, 3 fat stem cells, and 1 neural stem cell. There are a number of trials in various countries, Korea, China, and India, and unfortunately a number of times when you try and get access to some of these studies, it's difficult to demonstrate that they're being done in reputable places. That's not to be biased to those countries, but sometimes it's quite difficult to be sure that it's going to be reported and what it's aiming to do. However, there is one that appears to be quite authentic at this stage, and I think it's nearing completion from my colleagues tell what they've told me. And this is a particular company in Zurich, in Switzerland. It's a phase 1 study, and it's actually for thoracic, which is the chest part of your spinal cord where the people have had injury, and it's either complete spinal cord lesion or partial, and they're using a neural stem cell which came from a aborted fetus and is obviously a product which Stem Cells Inc. has, and so this is allogeneic, it's from somebody else, it's a foreign stem cell, and they've transplanted directly into the spinal cord at least 6 weeks after spinal cord injury. And the outcome of this study is to look at safety, not whether it's going to be beneficial or safety. There was another company called Neuron that some of you might ask me questions about in America, which was going to use a human embryonic stem cell for trials in spinal cord injury. And there was a great disappointment because the company actually stopped these trials because it wasn't considered to be financially viable for the company. Motor neuron disease actually is one of the most successful to date. And just by serendipity, I came across John Glass, who's a neurologist scientist at Emory University in USA, and he's actually just reported the first in human study, phase 1 study, using a fetus or embryo-derived neural stem cell, sorry, not an embryonic stem cell, but a later one. He had 12 patients, and half of them were people that were in wheelchairs and half of them not, and injected these stem cells into the lower part of the spinal cord and was able to demonstrate that it was a safe procedure. They actually injected 5 areas in the one side of the spinal cord in people that were not ambulatory, so they were in a wheelchair, and then they injected on both sides of the spinal cord in the lumbar region, and then moved on to people that were able to walk. So they did it in a graded manner to make sure that there wasn't going to be a major problem with these injections. This was reported towards the end of last year, and they're now moving on to what is far more important, and that is injecting or doing stem cell therapy in the neck, because that's where you have involvement of breathing and swallowing, and so that's critical to see whether that might be of benefit there. If you look on a clinical trials website, there are 10 human trials registered, but again, a number of them, it's unclear what their purpose is and how they're funded. 6 are using bone marrow stem cells, which aren't always clear how they'll be of benefit. So all of this leads me to, first of all, say that the whole field, I've become aware, talking with my colleagues around the world, is at this stage where we're seeing early trials in humans basically to ask a question about safety. Not yet how useful they are, because we have a lot of animal data to say they might be useful, but safety at this stage. That comes to this concept of stem cell tourism, and I would just say that I see a number of people around the world, from around Australia, that come to me and talk about their particular neurological condition and whether they would like to go somewhere and pay money to have stem cell therapy. There's the International Society for Stem Cell Research, which has a website that actually has 10 questions that you need to answer if you're thinking about this. And I think they're very fundamental to work out whether this particular place you might consider for stem cell therapy would be worthwhile. Unfortunately, I've got a number of patients that have been, their money has been taken from them with no benefit and, in fact, perhaps nothing given. The other aspect is that many times it's unclear that there is actually any proof that there's benefit with this stem cell. Many times it's patient commentary. There's no actually published data that would suggest that that's of benefit. Often you don't know where these stem cells come from, or indeed if they are stem cells, whether they have any infection. So many of the safeguards that are fundamental in Australia that we use may not be in place. And the other important aspect is that if you can see all the number of trials that have been done with a range of neurological diseases, if you go and go to a clinic or some country and have an unknown stem cell treatment, then that may prejudice you and preclude you from future studies, which I think would be a great pity because we're moving so fast in this. And many times the particular clinics will not follow people up, which again I think is a great loss and an indication that perhaps it's not being done correctly. So, in the remaining time, let me talk about my particular favourite stem cell from the tooth. So, we call them dental pulp stem cells, and they come from often young people that have had impacted wisdom teeth, and Gronthos and Sam Gronthos is a collaborator of mine since about 2002, and he and she found these when they were working in the States. She is a dentist, and he said there must be some stem cells in the tooth, and so they found them and published in 2000 that there were. They are what we call multipotent. They're not like an embryonic stem cell that can produce every type of cell type. They tend to produce only the cells that are useful for the tooth, but one particular one that's very useful for the brain is neurons. And they have a particular predilection to make brain cells, which is what's fascinated me about this particular stem cell population. They have a normal function in that if you damage your tooth, they'll try and put more dentine there. They'll actually, if you damage it so you can't feel something or it bleeds, they'll put more blood vessels in there and put more nerves for function. We've published a range of papers, and often the first in the world, demonstrating their what we call a neurogenic potential. That is how they interact and work as nerve cells, and I'll show you these. And also, we now know from another group and our own work that, in fact, a whole range of ages of people that still have their teeth have stem cells in their tooth. And you can see here some lovely photos. These stem cells actually grow on plastic quite well. And in fact, just recently, one of the people in my group, Rebecca Chu, has demonstrated even from a tooth from a 90-year-old person was able to show some cells that grew out. We don't yet know whether these are fully stem cells or whether they actually can produce neurons. We're doing experiments to that. But we do know that from other groups' published data that in fact they certainly appear to be multipotent and produce a range of other cells, but we don't know yet whether they also have the potential for neurons. We think they do, but we don't have that evidence fully Sheep and mouse also have stem cells in their tooth, so they're in varied ages and species. So the first studies that we did and published in 2008 looked at making brain cells from these stem cells from the tooth in a test tube. And this is very important because it means that they have a potential to do that for neurological disease. And these images just show you, I love neurons or brain cells because they have all these processes that look gorgeous under a microscope. And these images here just demonstrate that the one unique thing about a nerve cell is that it must be able to communicate with another nerve cell, much like humans. If you put a human by themselves, they're not much benefit. We do best as teams. And the same for nerve cells. And they're actually able to communicate electrically between each other. We then did an experiment which Rob would know about, which is an old embryological experiment using chicken eggs. And many of my neurological colleagues thought I'd lost the plot when I did these experiments. So if you take an egg and you make a window in it, so you can look down and see the embryo, and this is a picture of a very early chicken embryo. We took our human dental pulp stem cells that were labelled particularly with a gene that made them glow green, injected them by the brain, and then we waited 48 hours and were able to demonstrate that the green glowing cells that we knew had come from the human dental pulp actually were able to make neurons. And so we published that, and that was very exciting. More exciting was another finding which we had no idea that we would find. In fact, it wasn't a question, but we observed this. So sometimes some of the best excitement in research is seeing something that you weren't expecting. So this relates to wiring of the brain. So bear with me for a moment. So your face, where you feel, is innervated by a nerve called the trigeminal nerve. It has 3 branches, one to the forehead, one to your cheek, one to your chin. And you touch, and these 3 branches will feel and tell your brain that you're feeling things. So these 3 branches are here. One goes to the forehead, and these 2 go to your cheek and your chin. And this particular, we call this patterning of the nervous system. It grows out, and it's in all type of vertebrates with a spinal cord, and it's it's genetically programmed, and it's very hard to break this. So we injected in our human dental pulp stem cells, and you can see here a picture of the embryo. There's the head and the eye. And we found a remarkable event after 48 hours, where the stem cells from the tooth formed, then they produced a chemical signal which actually attracted the axons or the processes from the neurons to this particular area where the stem cells were. And this was the first evidence of neuroplasticity in the nervous system, be that in a chicken embryo, which you might say has no particular relevance, but I'll prove to you it does. And this also demonstrated to me this two-way interaction of stem cell and the nervous system, and that this is very different to just taking a drug and having a one-way interaction. So a number of these people, Agnes, Tanya, Wei Kei, and Kalia, who's got a... Eli still there, or he's gone? Gone, up. They helped me over a number of years to do a study which was fundamental, and that is to do with a stroke model. So we take a rat and we cause a stroke. I shouldn't say that though, should I, Sarah? And this is a particular form of stroke. This is a cross-section this way through the rat brain, and the white element here is where the stroke is. And this is, in fact, the most common stroke that I see in patients as a neurologist. It's called a middle cerebral artery stroke. And 24 hours after that stroke, we then injected in 2 places, one up high and one deeper down, about 300,000 of the human dental pulp stem cells. And we did a number of things which are very important in these studies, and that is that we tried to not allocate what animal received what cells, and we tried to blind all our researchers so they didn't know which animal were receiving what cells. So we had no hopefully less bias that many studies tend to have in this field. Then we did a number of tests, in fact over a month after we'd implanted these stem cells into the brain, behavioural studies. Many of them are similar to what we do with humans. There was a neuroscore, which is a bit like an NIH score that is used in humans with a stroke. So it's a battery of tests to look at how debilitated the animal is. Then a step test. I'll talk about the sticky paper test. That's a very simple one where you put a piece of paper onto the animal's forepaw, and obviously there's two aspects to this. One is it has to feel that the sticky paper is there and then try and get rid of it because it doesn't want it there. And then the fourth is an awful type of test where you take the animal and you rotate this rod and wait for it to fall off, which is a little mean. I won't talk about that. And then a ledge beam where you have this smelly box and you put the animal on one end and it runs up the box and you film how many footfolds it has doing all these. So the red boxes here demonstrate two studies that showed two of these assessments that demonstrated improvement in the forepaw, or like the arm in humans. One was a neuroscore, so you could basically see that in the vehicle or the control experiment where we injected in just some fluid that didn't have any stem cells in, the animals actually improved and then they plateaued out over a month, where in the animals that were treated, they continued to improve over the month period. In fact, they had their biggest improvement at 4 weeks. And the other improvement was in the forepaw, this sticky paper test. So at the beginning, when they had the stroke in this control group, you actually see that they spent no time trying to take the sticky paper off because they weren't aware it was there on their paw. And then they improved, but not very much, maybe to about 40% of the time they would try and take the sticky paper off, where you saw that there was almost up to 80 or 90% improvement with the animals after stem cell therapy. So we then demonstrated that these stem cells that we'd injected in the brain after a stroke had produced brain cells, but there were very few of them. In fact, we found an interesting aspect, and that is at 4 weeks, in 3 animals where we'd seen the most improvement in their function, we actually only had between 10 and 20,000 cells still alive that we could find, and we had certain ways of doing that. In fact, there were only 2.3% of cells that still existed at 4 weeks. So this takes me to Cambridge. This is King's College in Cambridge. And the question of how does this work? How does stem cell therapy in this rat model help to improve function? And the first is that this concept of replacement doesn't work, doesn't make sense in this stroke model. Because if you have a stroke of an animal of this magnitude, you've lost 1 to 200 million brain cells, and having 10 or 20,000 cells at the end of 4 weeks will not replace enough to make any benefit to that animal. So we then come to the concept of neuroplasticity. I had the pleasure of working with James Fawcett, who is the head of the Brain Repair Centre in Cambridge, and he's a world authority on neuroplasticity. And this is a wonderful image. It's an MRI of a human, and this person is being asked to speak. And we can actually, with magnetic resonance imaging, actually find the activated area of the brain that will work when you're doing a task. And in fact, there's a number of studies that demonstrate that if you have damage to the brain, that the brain is able to move around the area of the brain that's involved in that task. And so we're in collaboration to try and work out the mechanism of neuroplasticity that might underlie some of the results we saw in the animal model. And there's a number of exciting results that I can't tell you all about, but are very possible to lead to an idea of why we might see the benefit we do. So finally, dental pulp stem cells or stem cell therapy in stroke. So I've talked with you about the other neurological diseases, so I've left stroke to the end to not be too biased. So what is important to recognise, and we published a paper last year, a group of us, is that over the last decade there's a plethora of animal studies using stem cell therapy in stroke. In fact, there's 192 experiments, about 2,700 odd animals that have been used around the world over the last decade. And on average, we see about 40% improvement in function after a stroke in the animals. And these experiments with a range of different stem cells, from embryonic stem cells through induced pluripotent through to adult stem cells. They're injected either into the brain shortly after a stroke, called an acute stroke, or they're given in another way, whether it's into the brain or via the vein, in chronic stroke. That is when there's some weeks or months after the stroke in the animals. In fact, my study, or our study with dental pulp stem cells was one of the first to demonstrate this had a benefit in these rat model of stroke, and now that's being demonstrated by 4 other groups around the world that there's a functional benefit. So there's about 100 rats now that have been injected with these human stem cells from the tooth, and so far there appears to be no tumours and no side effects. In fact, adult stem cells, those from bone marrow, we know are exquisitely safe in regards to resulting in tumour formation. The more pluripotent that a stem cell is, like an embryonic stem cell, the less we have the ability to regulate it. We may learn how to do that in time, but at this stage probably not. Where adult stem cells with a much limited range of making different stem cell types doesn't have that same tumour risk. There's also in June this year a report from a group, Keith Muir in Glasgow, of a study from a company called Reneuron. This was a phase 1 study which was using a human neural stem cell. So this was taken from an aborted human fetus. The brain was removed and the cells were obtained and the stem cells were used, and they were genetically modified to make the cells proliferate, to make them keep growing, so that the company had a product. And they're called immortalized human neural stem cells. Then the study that was done was what's called a chronic stroke trial, and that is about 6 months or 60 months after a stroke, then the patients were injected into the brain with several million different amounts of these human neural stem cells. And to date, they've been looked at the patients for 3 to 24 months. And the major outcome of this particular study is safety. And they were able to demonstrate that, in fact, these cells were safe to date. One of the big problems of this particular study is that these are genetically programmed to keep proliferating. And as you might imagine, that increases their risk of tumour formation. And indeed, they only were able to use males, because with females, with the particular genetic abnormality or genetic programming they did, if a woman were to be given these cells and develop breast cancer, had a particular treatment called tamoxifen, that would perhaps switch on the gene to result in tumour formation. So there's a number of caveats with this particular study and this particular stem cell. However, it's the first in human that was done, and it provided many of us a lot of evidence about the safety of these cells, even though only 9 people were used. So what might happen in Adelaide? This is obviously going to change now. There's a bridge, I think, being built over it. So, and there'll be Port Power and Crow supporters there. So I'm proposing now that the time is appropriate for a tooth stroke study. In the clinical field, you often try and get an acronym that describes what you want to do. And so tooth, you can see I've been a bit poetic justice to pull out a number of letters to make tooth. And the idea is that this would be a phase 1 study. And it would be, again, a bit like the Glasgow study. It would be to look at patients who'd had a stroke previously, and at least probably 6 months, and that they have a stable disability. And the particular aspect of this study would be that we would consider having an autologous transplantation of dental pulp stem cells. So that is where we, if the person has their own teeth, we're able to take their tooth. And I must admit the dentists in Adelaide are enormously generous. They have given us a range of teeth, as I said, from a range of ages that we're now examining, from middle age, where I am, to older. And as I showed you, even a 90-year-old tooth, where we're looking to see whether they have this potential in a test tube, but we don't know what potential they might have outside of that. Be that as it may, this would be a study which would be done in Adelaide, and that it would allow us a 2-year follow-up, so people would have to live here, and then for life, because I plan to live here for a fair number of years, and I would follow up after that. The outcome of this, because there's so few patients, would be that this is a safety study. So it's too few, as I've tried to reiterate, to actually say whether there's benefit. But obviously, when you're having people that might consent to this, then you want to learn as much as possible, humanly possible, about what changes are occurring in the brain over that period of time. And so there are a number of possibilities that we might be able to do. One is that at the end of next year, 2014, there should be in place an MRI scan at SAMRI, the building that's being built down North Terrace, that will allow us to actually look at the function of the brain and how that would change. That's currently not available in Adelaide, so that would be a wonderful opportunity to look at people's brains after they've been given stem cell therapy. There are a number of challenges to this, talking with my colleagues around the world that have done some of these first clinical trials. It won't happen tomorrow. In fact, it won't happen the day after that. It will happen over the next 2 to 3 years just to get things started. And this is the first time that I've talked about this in a public forum. Funding is critical because it is expensive, and I'll be putting in a grant to this National Health and Medical Research Council, which is our major funding body. However, the Peter Couche Foundation will also be fundamental in raising funds for this study. We'll need to have patient advocacy, and so we'll be developing over the course of this year some focus groups where we will talk to stroke survivors to get their impressions of what the study they think are the limitations or benefits of this and what they would consider they would consent to. And certainly from some of my colleagues around the world, particularly in Cambridge, that's fundamental. Scientists and clinicians do not understand what the person goes through that's had a stroke or a particular neurological disease. The second is that there's a Therapeutic Goods Administration organisation in Australia, and that will take a couple of years to go through that paperwork to be able to demonstrate that we could do this in humans. And then an aspect called good manufacturing practice. And what that means is basically that we make a stem cell from a person's own tooth that is free of any infection and that is as standardized and reproducible as we can between one person and the other. And so that increases the likelihood of safety. And in fact, we have that already for patients that have bone marrow transplantations. We have what we call a GMP facility at the IMVS or SA PATH. Finally, this particular person, Peter Couche, who couldn't be here tonight because he's had a viral infection, and I said no, he didn't need to come. So hopefully this is podcast. Peter's, as a number of you would be aware, is one of the most remarkable human beings I know, because he had a stroke when he was 41, which was of his brainstem, which meant that he got completely locked in. However, he did develop some movement in his right index finger, and he's a very dogged type of person. About 5 years ago or so, he wrote me a number of letters to say, could I be involved with stem cell therapy with him? And I ignored these, and as he keeps telling me, and he can't speak, he writes. And then he suddenly appeared, he invited me to a launch of a book that he'd taken 6 years to write called Lifelines. And then I was interviewed by the ABC Australian Story because he had a story about his life and Simona, his beloved one. And he's actually formed a foundation with his colleagues, friends, I should say, in Adelaide, and they've raised to date almost $500,000, which has allowed my group to get to the point where we are now to consider actually to go forward in looking at a safety study. Peter wrote me a letter a little while back saying that he probably realises this won't be a benefit to him. I should say the other most remarkable person is both his wife and his brother. I'm not sure if Steve is here. Simona's probably not. So it's very critical to recognise how things occur. And as a clinician, you build up some resilience against the sadness of what you see. As a scientist, you want to pursue a particular line of action to make sure that things are correct. When I put on my scientific hat and talk with my science colleagues, they say, well, we don't know how it works, and that's true. And so we need to know a lot more. When I put on my clinician hat and I talk with my patients, then I realise the urgency of pushing this forward to try and understand, one, is it safe? And two, is it going to get through this valley of death and actually be something that will change people's lives? And whether it achieves that over the next several years, we will wait and see, but at least we'll be part of that picture here in Adelaide. So thank you very much for your time and attention. Yeah, I've got some... So there's... the forum is now open to questions, and we've got some people who will run around if you want to ask any. So please put your hands up.
Thank you very much. I have a personal interest. My father and his oldest brother died of motor neuron disease, and the middle brother died of a glioblastoma.
Yes.
But my question is, my mother's mother died of multi-infarct dementia.
Yep.
So just two questions. What about dementia, and what about... now I've forgotten that. the other motor neuron disease. I can't remember.
Yep. So in regard...
Hodgkin, Cholera, what's that one?
Huntington's.
Huntington's, yes.
Okay. So I didn't put in Alzheimer's disease. The reason is that we know very little about most neurological diseases. We know even less about Alzheimer's disease. In fact, one of the primary concepts about what underlies Alzheimer's disease is likely to be wrong, to do with amyloid protein, because a number of trials have failed in the last 5 years in regards to that. It's a disease actually of the whole brain, where the brain degenerates. And so at this stage, I am not convinced in the studies that I've seen in animals that we actually can make an impact on this disease. And certainly the clinical trials, I think, would be too soon. And I don't have a feel of how useful that would be. So I haven't gone to that point because I think that that's almost too great a task to overcome. The motor neuron disease I've talked about. There are a range of different forms of motor neuron disease. There's a familial form, which is 10% of people will have something that occurs in families. And we're finding out, indeed, a great deal about that particular disease using induced pluripotent stem cells. What I didn't tell you is that one of the most remarkable things now is that we can actually take your skin cell, someone, say, who has motor neuron disease, take their skin cell, do this trick with these genes and change it into an embryonic stem cell and make a neuron that would be, all intents and purposes, like the neuron of their brain or their nervous system, and study what's wrong with that neuron. And it's called modelling, disease modelling. And when I was in Boston just in June, there's a number of remarkable discoveries that are coming through in the scientific journals about what are the underlying mechanisms of some of these diseases, because it's very hard to get living brain cells from people. But if you can do them in this manner with these induced pluripotent stem cells, that gives us a great deal of knowledge and maybe new avenues to pursue. So it's not only therapy, but it's also modelling of a disease that leads up to other opportunities with stem cells.
Yeah, hi Simon, thanks very much for coming along and filling us with all this. We hear all these things through the press, but hearing it from the horse's mouth is great. Thanks very much. I had a stroke when I was 30 as a result of a migraine.
Yep.
Couldn't get to having my Panadol in time, and so I've got shadowing on my right side. The symptoms I had from that was, they were sticking pins and needles in my left side and couldn't feel it. I've since developed MS. I've had that for probably about 15 years. My auntie had MS and died from it, and my family have got autoimmune diseases.
Right.
But I don't know whether there's... I've only got about half a dozen lesions, and they've been stable. I've been in research with the QEH for Gilenia for the last 6 years, and I also did a trial with the CMAX as well.
Yep.
So I'm quite used to having, been poked and prodded for 15 years.
Yep.
But I don't know if that was something that could be used in your trial.
So, I mean, you're unlucky, obviously, greatly unlucky to both have stroke and MS. So at this stage, in regards to multiple sclerosis, Mark Slee, who's a neurologist at Flinders Medical Centre, has a very great interest in multiple sclerosis. He and I have discussed if there's this trial in Europe that we can be a part of, then we might consider being able to run a clinic or trial